at that, I'll hand over to Jens. He's a big group, so I'll let him do the intros.
Well, I got this one.
You have that one.
Yeah. So, well, thanks very much for the intro. I'm so happy to see so many faces that I recognize. I can't believe you come to listen to me again. I'm also happy to see so many faces I don't recognize. So I'm going to take credit for everything, but I'm obviously banned from the lab. So I'm doing the admin, and the rest of the people are sent, well, they're a little bit spread out. They're actually doing the real work. But it's a quite diverse group. So I'm going to go through some of the projects that we have that are ongoing. We have a secret project as well that I'm going to say a few words of. No slides for that one because it's really, really cool. But first of all, this is the lab group. So over here, we have Paula somewhere. Where are you hiding? Paula, hand up there. You should be here. I can't use the pointer on the TV. Is this working on the TV? No. All right, so we've got Paula here taking some environmental DNA samples. Environmental DNA is something we've really been focusing on since 2013. It's completely exploded, and everybody and his uncle is now wanting to do environmental DNA, which is great because we were one of the really early players in the field. Who else have we here? We've got Netan, Jens Carlsson, also known as Netan, and she's sitting over here. She's the lab manager and runs everything in the lab and all the finances and pretty much everything. Hand up, please. We got myself, Jens Carlsson. We got Michael. Michael was a star student. He won the prize for the best zoology student. So with us, of course. So we hijacked him. He's now a PhD student of ours. And we have Paul Flynn. He's the head of research and development over in Weatherby's. He's been doing a PhD with us since 2015. He probably will never finish, but we'd like to add his name to the crowd here. We got Robert Moyes, he's over here. We hijacked him from Italy, and he's doing a PhD here with us. And SJ, or Sergeant Power, she's not here at the moment. She just landed, coming back from Canada, so she couldn't make it here today, unfortunately. And we got Andrew Teig, who's it's a postdoc there, but I think we have the email now, so we can call him research fellow from very soon, 1st of May or so. Hand up, please. There we go. So that's the lab group, and you can see them spread out a little bit all over the place here. So we start with Paula. She is working on the project with ATU. It's an SFI-funded project. It's called GIDAS or JIDAS, or however we want to pronounce it. We haven't agreed how to call it. Nevertheless, she's working on gill disease. So that's something that affects salmon in farms. And our biggest task here is basically sequence the amoeba causing the disease. And the amoeba has something really cool in it. Let's see. So over here on the right-hand side, you can see gills that are being affected. And obviously on the right-hand side, you can see it's gone pretty far. So it's a complex disease. We're not really sure what's going on. The amoeba is involved. Is the amoeba being the invader, so to say, and open the gates for all the viruses and bacteria? What is the story? That's what we're trying to figure out. So this amoeba has a really cool thing. It has a eukaryote internal parasite symbiont, or whatever you want to call it. So that's really cool to have a eukaryote inside this amoeba. And its genome is reduced. That's the only thing we know. The amoeba's genome, it seems impossible to sequence the thing, but we're trying anyways, or rather Paula is trying anyways. And I just find this really cool from an evolutionary standpoint. People over in the salmon farms obviously want a vaccine and stuff like that, but from an evolutionary point, it's really interesting. But I'm going to move on here, see what else she's got in there. There's the Perkinsella, so I can't use the pointer. Back in the B109, we used to have a golf club as a pointer. I used to like that one. So basically, this is the amoeba, and then you have lots of different bacteria in here. It's kind of a microcosm of all kinds of biodiversity in there. And there you have the Perkinsella. So it has reduced genome, so it's obviously taking advantage of some of the genes in or some of the functions of the amoeba itself. So it's a really cool story. I'm trying to sequence the living daylight out of that one. So better understanding the disease development and of course get a vaccine at the end of the day. We're also doing an environmental DNA assay for this one. So we can go out to the farms and see when do you have this amoeba in the water? What can you do? What are the mitigations and so on? So if you want to know more about this very complex topic, Paula promised me that she's really, really happy to talk to you about it in nitty-gritty detail. So I'm going to move over to Robert and Michael. They're on a new project called Hydrogen. It's an EPA project, so it's EPA funded, and it's really looking into how this eDNA, environmental DNA, can be used in the Water Framework Directive? Can we complement some of the methods? They're very laborsome, the methods they're using in the Water Framework Directive. They include netting, for instance, where you basically kill the fish. It includes electrofishing, where you break their backs and so on. They include getting all the invertebrates with kick samples and so on and so forth. Lots of very highly invasive, laborsome methods. So can genetics do anything there? So we're trying to flog this a lot with using eDNA. And that's Robert and Michael. We just kidnapped them, as I said. There's a web page and so on. And this is, of course, with Mary Kelly Quinn. If you're working with EPA, Mary Kelly Quinn is always involved in there. A few people in here that I know is working with Mary Kelly Quinn and EPA. So this is Michael's part of it. We divided the basically 5 things in this project. So we're looking at fish, and that's Michael's thing. So looking at fish diversity, how do we assess that? Can we take water samples and sequence the 12S gene and say what fish are here? Can we quantify with it, et cetera, et cetera? It's also looking at diatoms. So that's the algae you find them all over the streams. And the type of algae, the type of diatom you will find in the river shifts depending on its nutrient levels. So that's also an indicator used by the Water Framework Directive. And we want to see if we can use genetics for it. This is quite difficult because the whole concept is based on knowing species. But if you ask people working with diatoms, no one knows what a diatom species is. They just have names and they're mislabeled and it's a mess. So it's not very easy, but that's what Michael is doing. Moving over to Robert, these are the sample sites, by the way. So it's a massive undertaking. We have 4, the ones in green here on the map. Can these guys see me on Zoom, maybe? The green sites here, there are intensive sites. We're going to visit them 4 times a year. And then you have the other sites there in sort of orangey. There are extensive sites. We're going to be visiting them once a year and do all the sampling that EPA would have done for the Water Framework Directive and IFI with electrofishing. And then we're going to go in and use genetic methods to get a comparison to see how well it works. So the lads, they're going to have a very, very busy summer traveling around to these places. Probably good fun. So Robert, he's in charge of the invertebrates, which is obviously a big thing in the Water Framework Directive. And he's also doing microbes, bacteria, trying to identify. It's kind of not really used by the EPA, but we're interested in detecting if there's effluents or whatever coming from farms or from humans. Can we identify the difference? Can we track sources using environmental DNA and bacteria? Because there should be different bacteria depending on what the source is. So that's in a nutshell, I believe, I hope. So the lads are really happy to talk about it in nitty-gritty detail again. There's another work program in this that we haven't brought up, and that's probably the coolest, is the blue sky aspect of it. So we did a little bit of back of an envelope calculation. Have you heard about the minion sequencing or MinION? The size of the sequencer is less than this. It has the capacity to do 22 gigabases, which is about 7 times the human genome over 72 hours. That's the capacity of the sequencer. So it's massive. I mean, look at the Hugo project, the Human Genome Project. It took years and years and years and cost them billions. This one costs about €1,000. We realized doing back of an envelope that we can barcode, so we get really nice barcodes for all Irish fauna. Well, how many individuals did we say we could do? A species? We're looking at every single eukaryote species living in Irish freshwater. We can do that in one run in 72 hours. So we got an Irish barcode. We got an Irish GenBank. That's really what we want to do. We can do that really, really quick for basically no cost. And we're looking into do that. The biggest cost here is probably going to be the DNA extraction, which used to be the cheapest part. It all changed. So that's one of the projects we're doing there. Moving over to Andy. Andy currently a postdoc. He's funded by a little logo there saying revive and restore. These are the mad people who are trying to revive the woolly mammoth, for instance. They're working on other things as well. For instance, charismatic megafauna like elephants. So Andy's involved there. He's also picked up an adjunct position over at Pwani University in Kenya, where we work. And it's a question about these elephants living in the forest. I think you got better slides here. So you see the Arabuko-Sokoke forest up in the left-hand corner there. That's the last remains of this massive coastal African forest that used to be on the whole East Coast. That's the last remains of it. And it's fenced in. And you can see in the lower left, the clear differences between the forest and where humans are farming. So that they're all the way up to the fence. So these elephants have been fenced in. And as you can see that in the middle one as well. Now, they don't even know how many elephants they have. Because if you look at the canopies or the canopy to the left, lower left, you can't see them. And in the summertime, well, when water is available, they spread out throughout that forest, and you can't see them at all. However, during the dry season, they must come out to find water. And there's only one year-round water hole. Do you have a slide of that? It's over down to the left there. So it's on an open field. It's a concrete well, basically, where they add water to it for the ellies to drink. It's the only water source during the dry season. So the concept here is that you can get, all elephants must come out ever so often and drink. So Andy's going to be over there, and he's already been there, as you can see, collecting shite, elephant shite. Elephant shite is actually very good shite. One of the things he also wants to look at is their diet, because the idea of reconnecting these elephants with the wild or the less fenced in savanna elephants, they need to have a corridor where these animals will move. And these animals will not move through or out of the forest unless they can find food. So what are they eating in the forest? We want to get that information from the diet in there and the remains in the shite, basically, and see if you can find those plants. But the main task here is counting elephants. And then what he's going to do is going to do a SNP analysis, basically doing what the forensic people do in the police forces all over the world, create a fingerprint, a DNA fingerprint, basically. And then he's going to see how many different fingerprints can he pick up in the shite. And that would actually correspond to the number of elephants that are out there, because each single one has to come to that water hole ever so often in the dry season. So on top of that, he's got a master's student. She's going to be a PhD student, as far as I understand. She's based in Kenya. She's looking at parasites. So the shit contains information on who you are, who you're related to. It contains information of what you're eating, of course, and it also contains information of your health status. So it's probably one of the best samples that you can get. It's good shit. Yeah, so it's currently developing SNP assays, and we're going to see if we can use these samples to identify individuals and related individuals. If you look on the larger scale, we have these poaching events where you have young elephants left alone. A question there again becomes, how can we reunite these? Where are the relatives of this lone baby elephant? And now they grow them up like the sheldricks. They have them in big farms, basically, where they let them grow up. And then they try to put them out. But if we can identify the great granny, the matriarch of her clan, her family, it's so much easier to put her out. Problem is we can get samples from the little baby, but the big elephants out there, we can't touch them because of all the permits that are involved. Turns out the CITES does not consider shite a tissue. So it's not covered by CITES. You can work with shite as much as we want. So it's good shite again. Anything to add there, Andy? Again, he's really happy to talk about this in very detail. Hope I didn't miss too much. We're also working elsewhere in Africa. We had Christina, who was, she's over in Zambia. She just defended her PhD, so she's Dr. Christina now. We're very, very proud of that. And it was together with Ag. And I've been invited to go down to Zambia and see if we can establish a pilot project to work on predators using shite samples again. And hopefully we will get a memorandum of understanding in the future. And we're going to be able to send UCD students to Zambia and do research fieldwork there. And at the same time, getting Zambian students coming over to Ireland. That's the sort of long-time hope. We hope to go down there and see what facilities they have to do environmental DNA and see if we can help them set up some facilities there. Very similar story in Namibia, where the target, I think the biggest target we're working with there is trying to work on rhino. They've got the white rhino and the black rhino, and we really want to work with them on non-invasive sampling. Michael here did his fourth year on rhinos in the Dublin Zoo, where he's proven now, and we have a fourth year student now, the proven that we can use footprint DNA. So we get a rhino footprint and we can get the DNA out of that and hopefully tell which individual it is. The extended use of the footprint DNA is that we've lost the northern white rhino. Thank you. I always say the northern black rhino. I get that wrong. The northern white rhino, there are 2 females left. What's his name? Sudan, I believe. The old man, the old bull that died a couple of years ago. So the 2 old females left. That's the only thing left of that subspecies. And the 2 old, and they can't carry any young. They're sperm frozen from Sudan, and these are his daughters. So that's a big problem there. However, South Sudan itself is a war zone. No one goes in there. But we have scouts in there, and they claim to found footprints of rhinos. So if those rhinos can be proven through environmental footprint DNA to be northern white rhinos, the species isn't gone. That would be a nice story, I hope. So in Namibia, we're focusing again on the white and the black rhino. The issue here is that they came back from almost extinction, and they're now out on farms all over Namibia. But we don't know the relatedness. They didn't have good stud books. So what's the story? Are they moving related individuals around and getting more and more inbreeding? What's the story? Similarly, it's also corridors. How do we get them to move around, doing diet studies and doing parasites, much like Andy's been developing the elephant studies. So we hope to go there. We have a memorandum of understanding that is waiting to be signed this autumn in Namibia. And we have the Irish ambassador to Namibia and Zambia is going to be there in person, we hope. And we're going to have the ambassador from Namibia who's in London. She's doing both Ireland and UK. We hope that she's going to be in Namibia to sign that off. So we hope that will be a really big event. More stuff in Africa. We've been down in Madagascar. We had a former PhD student. He's known, it's on TV now. It's running, what's the name of the show it does? No, he was on TV as well. Those, yeah, the Wild Sitters or something. He was working on Nile crocodiles in Madagascar. How the heck did they get there? Turns out that these are actually quite capable of swimming in salt water. So you're not safe if you're out on the beach there. So anyways, doing phylogenetics and looking at where the Madagascar crocodiles coming from. We've been over on the Ivory Coast where we had the manatees. We've been working with pygmy hippo and slender snout crocodiles. And those are all to the left, they're all environmental eDNA studies. So we've used water samples and trying to see where do we find these species. We have other things going on in Africa as well that might lead to something, but they're too early in development, so there's nothing much to talk about. We got a European Union project, one of those huge ones. You have, I don't know how many countries are involved, and it's impossible to manage it. And I'm so very happy I'm not managing this project. Imagine being the lead PI on an EU project. You must be bonkers doing that. So we have a small section, and it's really cool, showcasing eDNA capabilities across marine habitats. Showcasing, isn't that the best thing? You get to go out there and just show them, look what we can do. Isn't that funny? And you get to work with a really high-tech guy. So you got in the middle here, you have an AUV. So it's robotic. It's got a smart, it's kind of AI run. And so it's running around and sampling things. These guys can sample plankton. They can take all water chemistry and stuff. We are interested in plankton and water samples from an environmental eDNA point of view. So you're out in the ocean. You have these buoys or fixed stations where you can get water samples. We're currently working with a Norwegian group that up in Tromsø are working on a, what did they call it? A sailing skiff or something. So it's a robotic thing that's just on a skiff and it's got a sail, so and solar power. So you can be out there for months just sailing around. Takes some time before you get it back to land, but it has filtering capabilities. So it can filter water, put it in the cartridge, and then eventually you can get that cartridge back with an eDNA sample. So that's basically Nathan's biggest research project here. She's only half-time for the school, and this is 40% of her work is in this Marbethas, as it's called. And Tasman Crow, I don't know if there's anyone else here involved in Marbethas. They got a few people, yeah. So it's a big project, and yeah, I'm so happy I'm not leading it. This one is really, really disturbing. You see that GIF moving there? That's the pink salmon. Pink salmon came from the Kola Peninsula, south of the Kola Peninsula in the White Sea, where the Ruskies brought in for many years to try to get pink salmon farming going. They had problems to take hold. They started with even years. These are really cool because they are either even year spawners or odd year spawners. You can watch that GIF and tell me if these are even years or odd year spawners. So what happened? So of course, they took hold, an odd year spawner, and all of a sudden, as you see in the map there, look at 2017. Now we have in the northern Norway rivers, the world's best Atlantic salmon fishing rivers. We have more pink salmon than we have Atlantic salmon. This is a major problem. And as you see on that map, you have all this pinkish, of course, pink salmon, where they have been observed. So they are spreading like wildfire across Europe now. They've been observed to spawn in the River Nith in Scotland. We see them in the Moy pretty much every odd year. They will probably take hold in Ireland as well. We've seen them as south down as in France. They haven't made it into the Baltic, but they're knocking on the door. They made it all across the Newfoundland. This is a Pacific salmon. It does not belong in Atlantic waters. So this is a huge problem. Now, IFI, eventually, 2024, we have the funding. 2024, that's 7 years since the invasion started. That's how long it takes government agencies to respond. Finally, 7 years later, Inland Fisheries Ireland is spearheading this with NASCO as a main funder. NASCO is the North Atlantic Salmon Conservation Organization. Only now has NASCO been able to secure money from the European Union to do this. And there's a big project, as you can see, lots of different partners. We do not have staffing for this. So if you know anyone who's really good at eDNA, because this is going to be figuring out how we can use environmental DNA to track where the pink salmon are going. And if we could get early warnings and mitigation projects, like in Norway, they are basically shutting down rivers. They're back on using rotenone. They're building barriers to prevent the pink salmon from coming up. It's a massive, massive problem for those poor Norwegians who are lacking all that money. That was cynicism, by the way. So, yeah, I think I'm going to end there, but I've got one last story, and that I don't have any slides for because Andy forgot to put them in. And it's a really old project. It's called Beyond 2020. And it was running with Belfast, Queens. It was ourselves with DTU. Who else was it? Marine Institute. It's a large project looking at one watershed up in Mayo. And we got a lake core. Amongst all the different things we got a lake core. It's about 6 or 7 meters deep. And it goes all the way into the glacial till. And we scienced the life out of this one. So we took all the genetic markers we could come up with. We did CO1, we did 12S, we did 16S, and then shotgun the thing. Shotgun is just taking any DNA we found there without any PCR amplification. And it's starting to reveal a really nice story. Lately, we found Arctic fox in it. We found reindeer in it. We have some evidence that the red deer never went extinct in Ireland. We have probably oryx in there. And unfortunately, he found pike. That's a bad story. And for anyone working with fish, that's really, really bad. So we hope to get that published soon. And that's only on the mammal side, basically. You've been focusing, but there's so many more things in there. We have the bacterial, we have the invertebrates, and we have the fish, and we have the mammals. So it's going to be a massive undertaking. As I said, this is called Beyond 2020. The project probably started in 2018 or something, or 2017, because it was supposed to be a 3-year project. We never got the extension, which I thought would be even better. What better name than Beyond 2022? I thought that was brilliant, but we didn't get that funding. But yeah, it's been taking a long time, and the other PIs are going, why don't you publish? Why don't you publish? But there's no rule book for doing this, core. We're inventing ways as we go. So I find that one of the most exciting projects we have. No ramifications for management or anything, probably, but it's really cool. And I'll end there. Any questions?